All right, thank you everybody. Yes, I am indeed wearing two hats today. I'm representing Arakari Biosciences, which is commercializing technology developed in my lab at UC Irvine along with Steve George, now at Davis. I'm still at Irvine. My university makes me put this up. This is just to declare that I do have a potential conflict of interest, which they manage very carefully, I will say. Okay, so, all right, hang on. The slides on here aren't changing, is that okay? I can't see what's next. All right, okay, fine. So, what are the problems with preclinical testing? What are we trying to fix here? Well, what we know is that monolayers and monocultures do not represent in vivo conditions. Plastic that cells grow on is orders of magnitude stiffer than any tissue in your body, and that changes gene expression. All tissues that we know of are three-dimensional, so it's hard to model those in 2 dimensions. And we know that the geometry of cells does change the gene expression. A colleague in pharma has told me this phrase that I love: mice lie to us all the time. They're great for some things. The problem is they're not great for everything. And mice are not men. Their physiology is quite different in many different ways, and often that is overlooked to our detriment. And obviously there's a lot of heterogeneity lacking in mice. We also know from our work that drug responses are very different in 2D and 3D. We're going to see these data later. I just wanted to pop them up here. What we're seeing here is the growth of a tumor plotted on the y-axis over time in the control. And with this drug that we'll go through later, you can see we do very nicely blocking the growth of this tumor, whereas these same tumor cells in a monolayer are completely unaffected by this drug at any concentration. So the geometry really matters. So the technology that we've developed is the vascularized microorgan and vascularized microtumors. And what we're trying to capture here is the sweet spot between simple monolayer cultures and the definition of physiologic, which is the human, the patient. I've mentioned issues with mouse. We'll see there are issues with spheroids. So what we've created is a three-dimensional tissue that has its own vascular supply. So these tissues get supplied with nutrients through living blood vessels. This is your vascular system: a high-pressure arterial side, low-pressure venular side, and capillaries through which blood and nutrients are delivered. We schematize it this way with a diamond-shaped tissue chamber. We can put any tissue we like in here, and it will be supplied with blood vessels, living blood vessels, that connect these two microfluidic channels. We can put all of this into a 96-well plate. We get 16 individual tissues within a 96-well plate format, and you can see they grow quite nicely here. And this is one of our vascular networks. We label the endothelial cells so you can see them. And so here's the artery, the vein, and the capillary network. So why do we go to all this trouble to make blood vessels? Several reasons. As I mentioned, all organs are vascularized. All tissues get nutrients and drugs through living blood vessels. Vessels also help to pattern tissues. So we've learned this from multiple organs, that the very presence of the vasculature helps the differentiation of that organ. Most cells are within 100 microns of a blood vessel. And as we said, nutrients and drugs are delivered through the vasculature. So if we really want to understand a tissue and have a physiologic model of a tissue, we need to include the vasculature and have it perfused. So here's, if we can get the lights down at this point, if that's possible. Maybe? Not? Okay. So if you can see here, this is the artery, and you can see the thank you. You can see the capillaries coming off there. This is flow of blood through our device, through tissues in vivo. This is a nail cuticle. And in both cases, you can see there are areas of high flow, areas of low flow, and if you're close enough, you can see red blood cells squeezing through very narrow vessels here and here. So in these ways, we recapitulate the look and the function of blood vessels in vivo, and this is just looking at permeability, and what we find is the permeability matches what you would see in, say, capillaries in the skin, and this is 70 kilodalton size of albumin. Something the size of an antibody leaks out more slowly, as you'd predict. So that's the model, and what I'm going to show you are data on the tumor. So here we have in green a tumor growing in here with red blood vessels. We get stromal cells wrap around the vessels, and the vessels penetrate and grow in and around the tumor in green. And again, this tumor is going to get drugs delivered in a physiologic way. So these are data we've had for a while, but it's always good to show. This is growth of colon cancer in the device. Standard of care Folfox does a very nice job of blocking tumor growth. We can reproduce that in a spheroid to some extent. This is a triple negative breast cancer, again growing. Maybe you can see it grows more aggressively and invasively. It's controlled by standard of care here, which is Taxol. And here's the data for that. And, whoops, something's missing, but never mind. Okay, oh, there it is. So, too many buttons. We cannot grow MDAs in spheroids. MDA is highly invasive. They've lost adhesion molecules, which is why they're invasive, and they won't stick together and form spheroids. But as you can see, they grow very nicely in our device. So another reason to use these systems. We've grown multiple tumor types. We've yet to find one that doesn't grow, which is nice. And I want to show you some validation we've done with this and some interesting data that we have. So we did a comparison of tumor cells growing in monolayer in our device in mice and, in fact, in spheroids. And you can see here growth of the tumor in 2D is rapid. If you add drugs such as Folfox, you kill it stone dead in a couple of days. This is growth in our device and in a mouse. They completely overlap. If you add drug, you control the growth of the tumor, but you don't completely wipe it out. We did this with a second tumor type, same result, rapid growth in 2D, slower but similar growth in the VMT, our device in the mouse, and again, we can control but not completely kill the tumor in the device. When we did gene expression studies just on the tumor cells, you can see that the tumor cells in the VMT in the mouse look very similar, but completely different to those tumor cells in monolayer. The tumor cells in monolayer have a very different gene expression pattern, which is likely why they respond so differently to drug. So again, if you want to understand how a tumor works, you have to put it in an appropriate environment. So we've done single-cell-seq on this. We identify the different populations based on key expression of key genes. Here's just some grouping we did. Here's the monolayer, here's the spheroid, here's the VMT, and importantly, there's a large overlap between the spheroid and the monolayer, but the tumor is quite distinct. When we go through these data, and I'm just going to skip through it, this is published so you can see it, we can look at all the different tumor types, all the different stromal types within our device, and we can identify each one based on gene expression. Here we're just showing some genes, keratins and EpCAM, identifying the tumor populations within there. And then we can sort these standard methods. We get the various tumor populations here, endothelial populations. What we've done here is plotted how each of these subtypes distributes through the different geometries. This I find a little easier to explain. We've done it the other way around. We've taken the three geometries and said, just looking at the tumor cells, what's the dominant tumor type in each of these? So in our VMT, we find that tumor B is the predominant type, whereas in a spheroid, it's a completely different cluster, tumor A, that is hardly even present in our device. Monolayer is dominated by tumor C. So remember the VMT there. This is the stroma, and again, here's the monolayer and the VMT. and you can see the fibroblast B dominates in the VMT along with endothelial cell type A, whereas this different population of fibroblasts predominates in the monolayer. So, interesting. We've also mapped these data onto the same tumor cells growing in a mouse, and you can see they map very nicely. We relabeled them tumor A, tumor B, and a stem-like population. This is what we see in the mouse. This is the same in the VMT, so very similar again. And when we looked at the data, we found a strong TGF-beta signal, and so we decided to follow up on this, and we chose this drug, galanicidib, that targets the TGF-beta receptor. And so when we asked, does this work in our system, these are the data you saw earlier. Here's this drug in the VMT. At all the doses we tested, it's very good at blocking tumor growth. Here's the control. No effect in spheroid or monolayer. So we only see an effect of this drug in the VMT and in vivo. That's published work. And when we look at TGF-beta signaling components within the various cell populations, what was really interesting is this key gene that's involved in transmitting TGF-beta signaling inside cells, SMAD4, is completely absent in this tumor cell. That was kind of a shock. So we went to the literature and found that this tumor line is actually SMAD4 mutant. So that asks the question, how is it that this drug is so effective when there is actually no TGF-beta signaling going on through this pathway? Well, there is a lot of SMAD4 in one population here, which is the fibroblast B population. And fibroblast B, as you recall, is the predominant fibroblast population in our VMT. So we had a closer look at fibroblast B. It turns out it has a classic CAF signature, cancer-associated fibroblast, suggesting that this drug is actually targeting the stroma and therefore preventing tumor growth rather than targeting the tumor directly. Suggesting, again, if you really want to understand how a tumor works, you have to have these complex systems where these cell types can interact. We also looked at endothelial heterogeneity. I'll just go through this very quickly. We broke them down into these various groups. And interestingly, there's a population that appears in the presence of tumor, these blue cells, that's not present in a VMO. That's the vasculature without the tumor. So we would like to think these are tumor EC. And when we do a pseudotime plot, you can see the evolution, if you like, of our normal endothelial cells into this tumor EC population. So we're following up on that. Hopefully I've got a couple minutes left just to touch on personalized medicine. So our goal here is to take a patient who's got a colon tumor, extract that tumor or biopsy it, put it into our device, and we've done this, and here it is growing in there. And again, here's the tumor in our VMT device. We then want to do drug screening on this tumor and then provide that information back to the physician, who can then have a better chance of choosing a drug that might be effective against that patient. So this is the cycle that we would like to complete. We are around to this point. We are not currently approved to give data back to the physician, but we are generating data to suggest that we will be able to give useful information. So I'm just going to show you a little bit of that. These are patient-derived colorectal cancer tumors. Just some examples. Here you can see them growing in the device with the vasculature, so they grow quite nicely. Here are some examples. Here's growth. The black line is the control. This is in the presence of Folfox, the standard of care for colon cancer. And this tumor basically eats this stuff for breakfast. It has absolutely no effect from this patient. Galanicidib, the drug that we were talking about earlier, does appear to be effective. Here, Folfox does work on this patient tumor. Galanicidib slightly less well. Here's another example where Folfox doesn't work and Gal does. And here's just some other patients, and you can see we get variability. There's a lot of heterogeneity in how these patients respond, whether they respond to both drugs or one drug, or potentially none of these drugs. So we do know that geometry matters here. So we've also put these tumor cells into spheroids. Here's one of these patient tumors. Folfox has no effect on the growth of this tumor. Galenistatib works very nicely. Neither drug would show up in a spheroid assay. Here's another example. Again, Folfox doesn't work, Galenistatib does. In this case, the spheroid predicts that both drugs will work, whereas we would say they wouldn't. Interestingly, this patient had 5-FU, which is one of the major components of Folfox, and they actually progressed quite rapidly and died, we found out. And we believe we could have predicted the failure of that drug in that patient, and we might have suggested trying this drug, which we think probably would work. So we're continuing these studies, and we hope to get enough data that we can actually start providing useful information to clinicians. And one of the questions we always get is, how do you deal with heterogeneity when you're taking small biopsies? So this is some work that was done at Arikari. We took a tumor and made lots of little micro-tumors, and you can see them here. We then ran through T cells, and I can't tell you the details of those, let's just call them T cells. And we looked at extravasation of those. We counted extravasation, we counted how many of those were localized to these micro-tumors, and the ones that were closely localized are marked with yellow crosses here. We then looked at the growth of each of these tumors, and I've got 5 of them here, and we found that 3 of these tumors didn't grow very well at all. 2 didn't grow well, 3 grew pretty well. And when we correlated that with the infiltration of leukocytes, what we found is 2 of these tumors had very high levels of leukocyte infiltration. They were the ones that did not grow. 3 of the tumors did not recruit T cells very well, and those are the tumors that continued to grow. So this is really exciting data. It does suggest that we can capture heterogeneity, and that certainly in immuno-oncology settings, we can see how that heterogeneity plays out in terms of leukocyte recruitment and tumor killing. So to summarize, tissues and tumor microenvironments are complex, and if we want to understand how those work and to predict drug responses in those, we really need to use 3D systems that capture that complexity. Mice are not men or women, let's be fair, so they are useful in some circumstances, but are not useful in many, as we're seeing particularly in immuno-oncology, as it turns out. Vascularized tissues allow us to look at these interactions. We do know that vessels help to pattern tissues. That's important in understanding how those tissues work. And we're also understanding that targeting the tumor microenvironment can be a very good way of controlling tumor growth. And we've also now seen that tumors derived from patients can be well modeled in these platforms. So I've got 2 acknowledgement slides. A lot of the data you saw came from my UCI lab, and the tumor data were generated by Stephanie Hashi. A lot of the analyses were done by Chris Hatch. These guys are all working on different aspects of the body and how we can model it, all doing absolutely amazing work, which unfortunately I don't have time to tell you about. A lot of this is collaboration with Steve George, who's here, and Abe Lee at UCI and others, and supported, as always, by NIH, and thank you to them. And as I've got 2 hats on, this is our Arikari team, the people who sit in the office, the people who do the work. Thank you very much to those. And thank you. I'll be happy to take questions.